Okay. In the last class, we have learned few basic concepts of immune system, right? Today we'll be discussing disorders of immune system. Let us begin. So, there are various types of immune disorders are there. Few disorders are caused when our immune system is acting a little too much, so that it is causing damage to our own tissue. Then we call them as hypersensitivity disorders. Few diseases are caused when our immune system is not working up to the mark. So there is low immunity or there is nil immunity. We call them as immunodeficiency disorders. Okay. And the third category is autoimmune disorders. These are due to failure of tolerance to our own antigens. That is, our own immune system, it identify our own tissue as foreign tissue and it will start reacting against them. So these are the various types of immune disorders that are possible. Among these, we'll start our discussion with hypersensitivity disorders for this class. So, what is a hypersensitivity reaction? Let us understand this concept first. Hypersensitivity reaction is nothing but an excessive immune response to an antigen to an extent that it starts damaging our own tissue. Okay? Usually, when an antigen enters our body, immune system, it responds in a precise manner, in a very controlled manner, producing just enough immune response to clear the antigen. This is not an uncontrolled generation of an immune response. Okay, it is very much controlled. And once the antigen is cleared from our body, the same immune response, it has to come down also. And to attain this, there are certain control mechanisms are there in our system. Sometimes what happens is this regulation, this precise balance between the effector mechanisms and the control mechanisms is disturbed. So what can happen is there will be either an excess immune response or there is a failure of this control mechanism, leading to uncontrolled immune response which ultimately results in tissue damage. When such a thing is happening, we call it as a hypersensitivity reaction. Okay? Then what is meant by a hypersensitivity disease? So whenever a disease is a result of a hypersensitivity reaction, we call that disorder as hypersensitivity disorder. Simple. In simple words, in hypersensitivity disorders, the underlying pathogenetic mechanism is a hypersensitivity reaction. It is as simple as that. Okay? And depending upon the mechanism of hypersensitivity reaction which underlies the disorder, there are 4 different categories of hypersensitivity disorders are there. Starting from 1, type 1 hypersensitivity disorders, type 2 hypersensitivity disorders, type 3 hypersensitivity disorders and type 4 hypersensitivity disorders. For all these disorders, there are distinct hypersensitive mechanisms are there. That means that if you take type 1 hypersensitivity disorders, these are also called as immediate hypersensitivity disorders and the pathogenetic mechanism is very distinct from rest of the disorders. If you take type 2 hypersensitivity disorders, in these hypersensitivity reaction is antibody mediated. In type 3, it is a result of immune complex formation, that is antigen antibody complex formation. And in type 4 hypersensitivity disorders, these are almost exclusively caused by T cell mediated immune responses. Okay, so based on the underlying pathogenetic mechanism, these hypersensitivity disorders are classified into these 4 types. So for today's class, we will be discussing type 1 hypersensitivity disorders and type 2 hypersensitivity disorders. Okay, what is a type 1 hypersensitivity disorder? These disorders, they are also called as immediate hypersensitivity disorders, and in colloquial terms they are also called as allergies. These are actually rapid immunological reactions. That's why we call them as immediate hypersensitivity reactions. As soon as the antigen enters the body, the development of this hypersensitivity reaction is very rapid. So we call them as immediate hypersensitivity reactions. And these reactions, they occur in a previously sensitized individuals. What is meant by sensitization? Sensitization means the particular person, he has already experienced this antigen, okay, and now he is again being exposed to the same antigen. So what happens when he has encountered the antigen for the first time, he will have some memory of that antigen due to presence of memory T cells, right? When the next time, when the same antigen, when the person encounters, the development of immune reaction against that antigen will be more rapid. And particularly in type 1 hypersensitivity disorders, this reaction is even more rapid and it is even more potent. Okay, so these disorders, type 1 hypersensitivity disorders, they can be a systemic disorder or they can be a local reaction also. In these disorders, there are two well defined phases are there. The first phase is called as immediate reaction, and it develops within few minutes to few hours of exposure to the antigen. In this stage, the person suffers from all the features of vasodilatation, and depending on the organ system that is involved, smooth muscle spasms and edema, like that. But it subsides in few hours. And after a certain period of time, the second phase starts, which we call it as late phase reaction. And this late phase reaction is the culprit for the tissue damage. And this late phase reaction, it occurs without additional exposure to the antigen. There is no need of an antigen for this late phase reaction to start. After the immediate reaction has resolved, even without additional exposure to the antigen, this late phase reaction will start, and it is a sustained reaction. And in this reaction phase, there is infiltration of the tissue by several kinds of leukocytes, which leads to subsequent tissue damage, usually mucosal epithelial cell damage. This is how a type 1 hypersensitivity disorder works. Now, let us dig into step-by-step pathogenesis of type 1 hypersensitivity reactions and type 1 hypersensitivity disorders. So till now we know that type 1 hypersensitivity reactions, they will have an immediate phase and they will have an late phase reaction, right? How does these reaction phases are actually happening? What is the step-by-step pathogenesis? Let us look into that now. So the central cell that is responsible for type 1 hypersensitivity reactions is, remember, T helper cell type 2. T helper cell type 2. We also call it as Th2 helper cell. Okay. So what actually happens here is, few of the antigens that are present in the environment, for some unknown reasons, when they enter the body, they trigger a very potent T helper 2 cell response. We know that when a T cell when it encounters an antigen which is presented by an antigen presenting cell, depending on the nature of the antigen that is being presented by the antigen presenting cell, Th cells, they will differentiate into either Th1 or Th2 or Th17 type of cells. Few antigens, for some unknown reasons, they preferentially trigger Th2 type of immune response. And now what happens? If you remember from our previous discussion, we know that these helper T cell subsets, they have got their own signature cytokine set. Okay, for Th1 it is interferon gamma, for Th2 it is interleukin 4, interleukin 5 and interleukin 13, and for Th17 it is interleukin 17 and interleukin 22. These are called a signature set of cytokines, right? So now the cell that is being formed here is Th2 type of helper T cell. So what are the signature cytokines that will be released here? They are they are interleukin 4, interleukin 5 and interleukin 13. Once these are produced in the field, what do they do? Interleukin 4, it acts on B lymphocytes so that B lymphocytes, they will start producing a specific class of immunoglobulin called as immunoglobulin E. Out of all the types of immunoglobulins that B cells can produce, B cells that are activated by interleukin 4, they will secrete only immunoglobulin E. This is what we call it as class switching of B lymphocytes. Okay, and interleukin 5. This is specially designed to attract eosinophils to the field. That is the function of interleukin 5. And then we have interleukin 13. Function of this interleukin 13 is to activate the B cells that are producing immunoglobulin E. In other words, it helps interleukin 4 to improve production of B cells that are capable of producing immunoglobulin E. And apart from that, it also helps in increased mucus secretion. So, the signature cytokine set, interleukin 4, 5 and 13, they have got their distinct functions. Right. So, now what happens is, as a result of immunoglobulin E production, there are a class of cells called as mast cells. These mast cells, they have got a very high affinity for immunoglobulin E immunoglobulin. Okay, they have a receptors which have got high affinity for immunoglobulin E. And if you remember, mast cells are like powerhouses which store all the preformed chemical mediators in them. There are several kinds of preformed amines, preformed enzymes, preformed metabolites that are there, which are stored in mast cells. Okay, once the mast cell surface receptors, which have got high affinity for immunoglobulin E, once the immunoglobulin E comes and attaches itself to the mast cell, it will start degranulating itself into the surrounding tissue. As a result of that, all the cellular components, all the preformed mediators that are there in the mast cell, they will now be spilled into the tissues. Examples for these mediators are like histamine, which we also call it as tissue active amine. This histamine is responsible for increased vascular leakage, increased mucus production, and depending on the specific site of the tissue, like in your respiratory system, it can also induce smooth muscle spasm. And there are enzymes like proteases. They can directly damage the tissue just by digesting the tissue. And there are arachidonic acid metabolites are there, like all your prostaglandins, which are concerned with vasodilatation, vascular leakage and smooth muscle spasm, all which will lead into edema of the tissue as well as increased mucus production. And there is production of prostaglandin D2, which is specifically concerned with bronchospasm. So all these chemical mediators, they are responsible for the immediate phase of type 1 hypersensitivity reaction. Okay, then because of all these changes that happens in the tissues, there will be production of cytokines in the tissue field. Cytokines that can be produced here are tumor necrosis factor, interleukin 1 and interleukin 4. The function of these cytokines is they will invite all kinds of W bases into the tissues. This phase where cytokines are inviting all the W bases into the tissue field is late phase. Okay. Now all the recruited lymphocytes, all the recruited types of WBCs, not only lymphocytes but also eosinophils and other leukocytes, they will cause tissue damage by eroding the epithelial surfaces. Okay, so this is how an immediate reaction as well as a late reaction of type 1 hypersensitivity reaction happens. So remember, okay, let us look into the picture. All the story, it all begins with activation of a T cell which is differentiating into T helper 2 cell. Try to follow the diagram. So few antigens, due to some unknown reasons, when they are presented by dendritic cell to a naive T cell, it will differentiate into T helper type 2 cell. Okay. The signature cytokine set for these Th2 cells are interleukin 4, interleukin 5 and interleukin 13. Interleukin 4 causes B cell class switching leading to formation of immunoglobulin E. Interleukin 5 is responsible for attraction of the eosinophils. Interleukin 13 is concerned with further activation of IgE production and mucus secretion, right? So once there is formation of this IgE, what will happen is mast cells which has got high affinity receptors for IgE, they will bind with them. As a result of this interaction, there is massive mast cell degranulation which leads to production of vasoactive amines and some lipid metabolites and some enzymes that are released into the tissue, leading to tissue damage. And there is also production of cytokines which leads to late phase reaction leading to tissue damage. Okay, let us see the. So there are several very good examples for immediate hypersensitivity disorders. Very important clinical syndrome is anaphylaxis, which is characterized by fall in blood pressure, which is due to a result of vascular dilatation, and there is airway obstruction due to laryngeal edema here. Okay, and one more very good clinical example for type 1 hypersensitivity disorders is bronchial asthma. Here the airway obstruction is caused by bronchial smooth muscle hyperactivity, and there is inflammation and tissue injury caused by late phase reaction. Okay, and there are allergic rhinitis, sinusitis and some food allergies. They are also very good examples for type 1 hypersensitivity disorders. So the bottom line here is, in all these disorders, anaphylaxis, bronchial asthma, allergic rhinitis, sinusitis, food allergies, whatever is the disorder, the underlying pathogenetic mechanism here is type 1 hypersensitivity reaction, which ultimately leads to tissue damage, which projects as a disorder. Okay, right. Now let us talk about type 2 hypersensitivity reactions. These type 2 hypersensitivity disorders, they are also called as antibody mediated disorders. So the underlying pathogenic mechanism here, it involves antibody mediated reactions. That's why we call them as antibody mediated disorders. Here what happens is, the antibodies, they react with antigens that are present on the cell surface or which are present on the extracellular matrix also. Try to understand the location of the antigens here. Here the antigens are either located on the surface of the cell or they are present in the extracellular matrix. Whatever may be the location of the antigen, these antibodies, they will go interact with antigens leading to cellular dysfunction ultimately. Okay, so there are three different mechanisms through which the antibody mediated immune reactions can cause disorders. Okay, but the first one is by destroying the cells itself. Okay, by mechanisms like opsonization and phagocytosis. Here what happens is the antigens, when they are located on the cell surface, these antibodies they will go coat that antigen, which finally leads to opsonization and phagocytosis of the particular cell. Or the second mechanism is they can trigger the inflammation. Antibodies they can trigger the inflammation by complement mediated inflammatory reactions. Here the antigen is usually located on the extracellular matrix rather than on the surface of the cell. Once the antibody goes and identifies an antigen which is located on the extracellular matrix, this combination it can trigger complement system, which leads to inflammatory reaction. Okay, and the third mechanism is sometimes these antibodies they can go and directly block few substances, thereby they interfere with the normal function of that system. This we can discuss later in depth. Now let us begin destruction of the cell by opsonization and phagocytosis. Let us look into this picture. So try to look at the picture first. The first mechanism here is opsonization and phagocytosis. We all know what is an opsonization, right? So it is coating of an antigen with a appropriate antibody. When an IgG antibody, when it coats an antigen, phagocytes they have got specific receptors for IgG antibodies. Okay, when a cell which is coated with IgG antibody, it is an easy prey for phagocyte. Okay, not only identification by the phagocyte, when an IgG or IgM antibodies are present on the cell surface, they can also activate complement system. Okay, so once the complement system is activated, there is subsequent production of different complement system activation components like C3, C5a, all these byproducts are produced here. Okay, uh, phagocytes they also have got high affinity receptors for these byproducts also. Okay, so by these two mechanisms, phagocytes they can easily identify opsonized cells and phagocytosis can happen. There is one more mechanism called as antibody dependent cellular cytotoxicity. So here what happens is cells that are coated with IgG antibody, they are directly killed by a variety of effector cells, especially NK cells and also macrophages. So these cells they bind to the target cells with their receptors for IgG Fc fragment, and the cell lysis will be proceeded even without phagocytosis. Okay, so this is what we call it as antibody dependent cellular cytotoxicity. This is the first mechanism here. And then the second mechanism is antibody mediated inflammation. So what happens here is once the antibodies go and they attack the antigen that is present on the extracellular matrix. Complement system is activated here. So as a result of complement system activation, there is production of several byproducts like C5a and C3a, which act as potent chemotaxins. Okay, so they invite several other leukocytes to the site of antibody deposition, and these invited leukocytes they will start destroying the tissue by activating an inflammatory process. Okay, and the third mechanism is antibody mediated cellular dysfunction. This is an interesting mechanism. Here the best example we can take here is disease of myasthenia gravis. Okay, so myasthenia gravis is a neuromuscular junction disorder. Usually the neuron ends of the neuron they will store one neurotransmitter called as acetylcholine. On proper nerve stimulation, this acetylcholine will be released into the extracellular surface, and this acetylcholine is supposed to act with its receptor that is present on the muscle membrane. Okay, this interaction will lead to a synapse. A synaptic firing. Okay, so what happens sometimes is there is an antibody production against this acetylcholine receptor that is present on the muscle. So they competitively inhibit binding of this acetylcholine with its respective receptor. So what will happen now? Even if there is a nerve stimulation and subsequent acetylcholine release, as acetylcholine is unable to react with its respective receptor, there will be blockage of that stimulus. Finally, it will manifest as a disease. Okay, and the similar example that we can talk about here is uh, Graves' disease. Okay, here the antibody is directed against thyroid stimulating hormone receptor. Okay, right. So these are the different mechanisms by which antibody mediated reactions can lead to tissue dysfunction. One is opsonization and phagocytosis, other one is complement mediated tissue inflammation, and third one is antibody mediated cellular dysfunction. The best example for opsonization